Welcome to this episode of Foundry Fireside. My name is Tiffany Chen, and I'm an intern on the Foundry Communications team. Today, I'll be chatting with one of the Foundry postdocs, Harika Dechiraju. Welcome, Harika, and thank you for joining us. Could you please introduce yourself?
Thank you, Tiffany. So I am a postdoc working with Dr. Aeron Hammack in the Nanofabrication facility at the Molecular Foundry. I also work across the Biological Nanostructures facility and NSEM. And my research focuses on building nanoscale biosensors and digital microfluidic platforms that can detect biological molecules with high sensitivity using very small sample volumes.
Thanks, Harika. What exactly are nanoscale biosensor and microfluidic platforms?
Nanoscale biosensors are tiny devices that convert any kind of biological signals or interactions into measurable, let's say, electrical or optical signals. And because the sensing elements are so small, they can detect very, very subtle biological changes that might otherwise be missed. And coming to microfluidic platforms, microfluidics allows us to precisely manipulate small amounts of liquids through microscale channels. And it also helps us automate processes such as mixing and transporting these samples. And so when you combine microfluidics with nanoscale sensors, we can perform very sophisticated assays using just a fraction of the samples or reagents that are required by traditional lab methods.
That's very interesting. How do you make a biosensor?
So, building a biosensor is a very interdisciplinary process. We usually start by designing the sensor element itself that is sensitive to the biological targets of interest. And then we fabricate it in the clean room using nanofabrication techniques, which are very similar to those used by the semiconductor industry in semiconductor manufacturing. And so by using these techniques, we can make structures that are only like tens or hundreds of nanometers in size, so like really, really small. Finally, we integrate the sensor with external electronics or characterization systems and sometimes microfluidic systems that can flow biosamples in and out of the sensing area. And finally, that's how we have a biosensor that can convert a biological signal into another measurable signal.
Adding on, how do microfluidic platforms miniaturize and automate biological assays?
So, traditional biological assays often involve just manual pipetting steps and relatively large sample volumes in the milliliter scale. Microfluidic platforms really shrink those operations into very tiny channels that are, micron scaled. And what we can also do with these platforms is we can program them to automatically perform all of the mixing or the sample preparation and sensing steps onto a single chip. And so this can reduce human error, it shortens experiment times, also reduces the amount of reagents we use or the plastic that we consume to run these experiments, and makes them more reproducible in that way.
That's so cool. Moving on, what does a day in the life look like for you?
It really varies. Some days are very lab intense, where I'm in the clean room all day or I'm at the microscope imaging and taking data. Some days it's more chill. I read papers or I analyze the data that I have. We have meetings to discuss how the project is going and to come up with new ideas and new experiments to run. So I would say no 2 days are the same. Every day has something different.
Thanks for sharing that. I understand you have a bachelor's in instrumentation and control engineering and a PhD in electrical and computer engineering. Why did you choose engineering?
Engineering appealed to me because it's a mix of analytical thinking and creativity. So you learn how things work, but you also learn how to build them and make solutions for real world problems. And as I was going through my bachelor's degree, I realized that a lot of today's biggest healthcare challenges can benefit from engineering approaches. And that kind of motivated me to apply my engineering background to bio applications, which then led to my PhD and me being here.
Adding on, How do you apply engineering principles to biological problems in your research?
I think of engineering as something that helps us develop tools that can make biology easier to study. And so we can make devices that precisely control fluids, detect very small biological signals, and generate some kind of quantitative measurements that would be very difficult to obtain otherwise. So I believe engineering really complements biology in a way that it gives researchers new ways to observe and understand the biological processes.
That's so interesting. Why does this area of research excite you?
I really enjoy working in interdisciplinary fields. For example, now I'm applying semiconductor manufacturing techniques to make devices for biological applications. And I find it very rewarding because in these fields, we have the maximum scope for innovation. And it's also because the advances in engineering can enable discoveries in completely different fields. And so while building a new sensor is like a small technological achievement, it can help biologists perform experiments that just weren't possible before. And so even if it's a small thing that I can contribute, it can make a big impact. And that really excites me.
That's great. How do you think your research will make impacts outside of the lab?
I hope that the technologies that we are developing here can become tools that other researchers outside the lab can use to accelerate scientific discovery. Better measurement tools can improve, how we study cells, how we understand different mechanisms that are going on, and develop new experimental techniques. And so, even though a lot of the work that I'm doing is fundamental research, it also has the potential to influence areas like healthcare, environmental monitoring, or biotechnology as the technology continues to mature.
That's amazing to hear. What is one upcoming project you're excited about?
So I'm currently working on making these nanofluidic devices with sensors integrated into them, where we can flow biological analytes and sense them at the same time in situ in a TEM. And this is pretty interesting because doing such liquid-based experiments in a TEM is very challenging, and not a lot of people have done it for the application that I'm trying to do. So I'm really excited to see the results that we get from this project and the insights that we have.
That sounds so exciting. What is your hope or ultimate goal for what you would like to achieve with your research?
I think throughout science, there's usually big breakthroughs that kind of follow instruments, right? So once the microscope was invented, it opened up a whole new area for understanding the cellular world. Uh, Sequencing opened up how we understand the genome. And my hope is also to add something to that lineage, maybe a device or a platform that can let researchers observe biology in a way that they couldn't do before. And if any of these tools eventually make their way into like better healthcare technologies, that would be the most rewarding outcome I can hope for.
All right, that's all the questions I have for today. Thank you for joining us. It was really great learning about your research. For those of you watching, if you're interested in learning more about research at the Molecular Foundry, stay tuned for more episodes of Foundry Fireside.